Okay, so what we're going to do is A equals T, right? But in RNA, T is U. And then we're going to have T, which is A. G goes to C. A is U. U, 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 G, U, U, C, C. T is going to go A. A, U. You got me there, right? The reason that we're putting the U there is because in DNA it would have been T. T is not present. We're going to replace that with a U in RNA. In the nucleotide, that is 3. So when it says to make these proteins, UAC, U, 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 G, U, U, C, C, A, U. You see how we're coming up with each 3 of those because those are considered nucleotides. These code for a certain protein. All right, now I'm going to go on down because I want to show you how to use the chart. This is the chart that you're going to go to. So for instance, if I have U, 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 U is my nucleotide. I'm going to work from the inside out. The first letter is going to start at the biggest one. All right? And then I'm going to go U. I'm going to go to my next biggest box. Now, whatever my third letter is where I'm going to code for phenylalanine. Now, what if I had UUC? It would still code for phenylalanine. All right, let's do another one. Let's say I have CAG. C. Here, I'm going to go to my A. And then G. I'm going to code for glutamine. I'm going to do one more. UGA, University of Georgia. No, I'm just kidding. Okay? UGA. Now, you can do this for any. If it starts with an A, you're going to start at the A. Okay? Actually, yeah, the one example I was going to do. UGA. U, G, and A. This is a stop codon for polymerase. Polymerase, anything that ends in ASC is an enzyme. Okay, so polymerase is an enzyme that helps to copy genetic material. So what a stop codon says is at this point right here, we're stopping. It's called a stop codon. Okay, so when you are answering that question of where you converted your A's, G's, and C's, and you put them in your nucleotide sequencing, that is how you come up with following to fill out your chart. Now you may have one, it's kind of like, what's that game, Sudoku or wherever you have like, you're given part of the information in your chart. And you may have to work forwards or backwards in order to figure out what you're coding for, is what I'm trying to tell you. talking about right here. Now, when you go and you look and read all this, this is going to explain your DNA triplet, your messenger RNA codon, transfer RNA anticodon. So here is what I'm saying. You're not given every single one of them. You may have to work forwards in order to get to this. Or you may have to work backwards to get to that. Alright, any questions on that? You'll have to go back and read some of that. You definitely want to go back and read that area you're talking. I just wanted to give you a little heads up on that DNA. That way you didn't get stuck right from the start. Okay? Now, the last page, you're going to go and you're going to do a virtual. It's called a fishbowl. Alright? And you're going to define biological evolution. What is the difference between microevolution and macroevolution? What are the 5 conditions that must be met for the proportions of alleles to not change? Hardy-Weinberg equilibrium, we're going to use this as a null hypothesis. If you remember, a null hypothesis says if we do this, then it will not have an effect on this. That's a null hypothesis. Okay? And I think you've got 5 questions there. Okay? So you're going to upload pages 7 to 14 for your homework. Any questions?